Thank you. Thank you for this very nice presentation. Hello to everybody. I am happy to be here today to speak with you about some of our work that we are carrying at the University of Pavia, dealing with seed priming and the molecular approaches that we are applying in order to understand better how it's working. So before start, I'd like to thank both the ISSS and ISTA for this opportunity, and thank you all for being here. So I have organized my talk into two parts. Let's say in the first part, I will give just some brief overview on the seed priming research and some key, let's say, concepts related to the seed pre-germinative metabolism and how the DNA damage response is involved in this. While in the second part, I will speak more about the system that we have developed to investigate seed priming at the molecular level, following kind of the main issues related with these techniques that revolve around its high variability, time to stop or to apply and to stop the treatment, and the identification of some early markers of early efficiency. So just a couple of weeks ago, I came across this work that is actually quantifying the trends in seed priming research for the past 30 years. Let me put my laser point, okay. Using the articles that were deposited on the Web of Science, and I think it's very nice to see how the trends, how the number and the interest in this research has exponentially increased during the past couple of years. Also looking at, let's say, keywords that are mainly or preponderantly present in this research, we can see how this is applied to different types of seeds and different types of stresses, including multiple processes that are being looked upon. And this paper also nicely shows the interaction that is being formed throughout the research community throughout the world, and the high interest, again, so how we collaborate all together to improve the knowledge and the application of these techniques. So spending a few time on the seed germination, we all know that this starts with the imbibition phase and ends with the appearance of the true first leaves when the plantlets are then able to perform photosynthesis. But the pre-germinative metabolism is more temporarily condensed, and it actually ends with the radical protrusion phase. And during this time, there is a high increase of metabolic and transcriptional activities that are induced by this by the water uptake in the seeds, which we know activates respiration, and with the activation of respiration, there is the production of reactive oxygen species. And thanks to the famous Bailey's oxidative window concept, we know that these molecules have an important role to induce the activation of signaling pathways that lead to the progression of germination. But if certain levels are overpassed, this induces damages to different molecules, including the DNA. So for a seedling to develop normally, these damages at the level of DNA need to be properly and rapidly repaired before the start of cell division, and this happens with the activation of the DNA repair system that are controlled by the DNA damage response, or the DDR pathway. So what is DDR? We can define it as an evolutionary conserved signal transduction pathway that is formed of different sensors like the MNR complex, transducers, the ATM and ATR kinases, and effectors, that is the implants, is the suppressor of gamma response 1. So SOG1 is the master regulator of downstream processes that include DNA repair, endocyte reduplication, program cell death, and cell cycle checkpoints. So what generally happens is that after sensing the damage, the cells activate the DNA repair pathways while concomitantly blocking the cell cycle progression in order to allow this repair to properly to happen. But if the damage, let's say if the damage is too much for the repair process to be effective, cell can choose to go through programmed cell death. So during plant development, let's say that the DDR, we know that DDR is mostly active during the first phases of development to avoid accumulation of DNA damage, while its activity slowly decreases towards senescence, where DNA damage accumulates at higher levels. But what happens in the seeds? So we know that in this system there are different phases where DNA damage can accumulate, and this includes maturation and early germination due to this rapid loss and uptake of water, but also during seed storage, especially when the conditions are suboptimal. While the repair process actually happens only during the pre-germinative metabolism in the second phase of the germination, as you also saw in the previous slide. So much of the work on seed DDR actually comes from some of our very nice colleagues from Leeds University that studied this process, especially during seed aging and mostly related to the cell cycle checkpoints and the programmed cell death, while in our work we focused more on the repair processes during seed germination and genotoxic stresses, as well as during seed priming. So we know that the term priming can be actually defined as an adaptive strategy where exposure to one stimulus can influence the response to subsequent stimuli, therefore activating this physiological state that enables plants to respond more rapidly to stress. So when we apply these to seeds, we speak about pre-sowing treatment, which boosts seed germination and the response to stress as well. So we can generally classify seed priming into three main, let's say, techniques or methods that involve chemical, physical, and biological treatments. Within the chemical ones, the chemical ones are mainly or often based on the use of aqueous solution and follows this so-called rehydration-dehydration cycle, where the first step is seed imbibition for a certain amount of time, followed by seed dehydration, and then re-imbibition following post-priming up to start of the germination. So as I was telling you before, this rapid water loss and water uptake and the activation of the metabolism leads to the accumulation of ROS and production of oxidative damage. And to limit and repair this damage, the seeds need to have a very highly active antioxidant response and DDR response. And what we saw during time is that actually high quality seeds are defined by more efficient processes in this sense. So how did we arrive to see this? Is that during the past 10-15 years, we have developed a different system in order to investigate the priming at the molecular level. So for this, we have looked at different time points, starting from dry seed to different hours during the imbibition curve, but as well as during the dry bag. We looked at the radical protrusion and the seedling growth, first stages of the seedlings. And to this, we apply the different treatments and different analyses that allow us to have this, let's say, global overall view of what's happening during seed germination. So some of these treatments, like sodium butyrate and trichostatin A, serve as stress agents, while others like kinetin, sodium chloride, and polyethylene glycol can serve both as priming and stress agent, depending on the concentration and the time of application. Among the analyses that we are performing, we have what we like to call this basic toolkit, where we start first with a thorough phenotyping, then we go to measure the levels of ROS that accumulate or that they are released from the seeds, as well as quantifying the accumulation of damage and the DNA repair kinetics, along with monitoring the expression of genes involved mainly in antioxidant and DDR, but also in other pathways. Then we build up on this basic analysis with more in-depth and high-throughput ones using, for instance, ultrastructural transcriptional electron microscopy, HPLC, and different type of omics. So we have applied this mostly to our species of interest is mainly the model legume Medicago truncatula, but lately we expanded this work to other legumes as well as to cereals and horticultural species like eggplants, tomatoes, or lettuce, and even to poplar trees. So in the next slide, I'm going to show you some of the results that we got throughout the years, starting with what I here generally called the chemical stress system. So in this first work, we have used trichostatin A, or TSA, as a stress agent, because this agent is known to induce chromatin rearrangements. And we have monitored the response to this treatment during germination, radical protrusion, and seedling growth. And with the phenotyping analysis, you can see that both doses that we use in this case are able to inhibit the progression of germination as well as the seedling development. And this was correlated with dose-dependent accumulation of DNA damage both in the radical and seedlings, and upregulation of some genes involved in the base excision repair system, like atoxoguanine glycosylase liase 1, ligase 4, and the tyrosyl DNA phosphodiesterases. Then we looked at the antioxidant response and see also here some enhancement in the radical, enhanced activity in the radical and seedlings, coupled with upregulation of ROS scavengers like superoxide dismutase, ascorbat peroxidase, and metallothioneins. So given the effect of TSA on chromatin decondensation status, along with the fact that we see growth inhibition in these seedlings, we also looked at other genes that are involved in the chromatin remodeling process or in cell proliferation. And also here we see that these genes are differentially upregulated depending on the dose and the type of tissues that we are using. But what we wanted to see next is how these processes are correlated to one another. So for this, we have performed this correlation analysis using all the different types of measurements that we performed in this study. And what we see is this highly positive correlation between, for instance, the TRAP gene and OGG1, which reinforce the link that exists between DNA repair and chromatin remodeling, also given the fact that the repair machinery, in order to access the damaged site, it needs to have, the chromatin needs to be decondensed. So the chromatin decondensation status influences the efficiency of the repair systems. While, on the other hand, the correlation between OGG and SOD and APIC genes reinforce the link between DNA repair and antioxidant response, mostly mediated by the reactive oxygen species. So this work shows us that the TSA is able to block the cell cycle, induce ROS accumulation and DNA damage, and all these events can touch directly or indirectly molecular processes that contribute to the seed stress response. Another agent that has similar effects to TSA is sodium butyrate, a short fatty acid with histone hyperacetylation activity, And again, we used this agent during seed germination. We applied it during seed germination, but in this work, what we did was to couple the DNA damage accumulation and activation of their systems, coupled it with a non-targeted metabolomic approach. And by looking into the results of the differentially accumulated metabolites in this system, we observed that mostly at the radical protrusion phase, there were some interesting players like metabolites that are involved in polyamine biosynthesis, uracil degradation pathway, and purine and pyrimidine metabolism that could be linked to this, what we like to call the metabolic hallmarks of the seed response to genotoxic stress. So this metabolomic approach we also applied in another system where we have used kinetin, that is, a well-known growth hormone, growth inducer hormone that can be used both as priming and stress agent. So why so? Because we saw that when we applied the kinetin only during imbibition, this resulted in accelerated germination, while if the treatment was continuous, this resulted in inhibition of plantlet growth, especially at the root level, and consequent accumulation of DNA damage. While in this case, the results of the metabolomic analysis especially during seed imbibition, show that the observed changes were associated with fast metabolic depletion that corresponded to the observed fast germination. However, at the radical protrusion stage, some of these metabolites, like for instance spermidine, was an indicator of stress, and it corroborates also the data that we obtained with the NAB system. So what we learned from this first work, or these chemical stress systems, have allowed us to identify some quality or stress hallmarks that are activating during the early stages of the seed pre-germinative, the seed germination, because we are working specifically on the pre-germinative metabolism. And these hallmarks can be both DDR and antioxidant genes, as well as different type of metabolites, so broadening the type of analysis that can be performed in this sense. And among these metabolites, for instance, the polyamine are related to antioxidant stress, while dehydroxyuracil can be envisioned as the metabolic signatures of DNA damage. So going to priming, we know that one of the, let's say, challenges that are related to the development of efficient protocol is to know the time to stop the treatment, because the prolonged exposure to these treatments, or what we are calling here as overpriming, lead to the loss of desiccation tolerance. So what we did here, we wanted to try to identify some early indicators that can allow us to understand when the desiccation tolerance starts to be lost. And first, we developed this hydropriming, this priming over priming system based on using two different Medicago species. And in this case, we used both hydropriming and hormopriming with kinetin that were applied for 2 hours or for 24 hours. And we see that after 2 hours, we have development of improved germination, development of normal seedlings, while after 24 hours, the seedlings, we can define, or we are referring this to the formation of abnormal seedlings, so not proper germination. Then we went to measure the levels of the reactive oxygen species during both the imbibition and the dry bag phases, as well as the radical protrusion stage. We've done this by using specific fluorescent probes like dihydrofluorescein diacetate and double histochemical staining for the radical protrusion. And as you can see, the overprimed tissues are characterized by very high accumulation of these reactive oxygen species. Then we have applied these methods to these methods to measure ROS, also to other species like soybean, tomato, and wheat. And what I just want to point out here that these are non-invasive methods that allow us also to follow the germination progression after the measuring of the ROS levels. But within this priming over priming system, we also wanted to understand a bit which are the reasons behind this loss of desiccation tolerance. So we went to look comparatively in between prime and over prime seeds specifically during dryback, and applying our analysis, we again saw the accumulation of DNA damage, especially during over priming, coupled with differential expression of genes acting as DDR sensors or effectors. What we saw is also differences within the nucleolar architecture that were coupled with an altered ribogenesis observed through looking into the expression of genes involved in ribosome biogenesis, but also we also monitored the ribosomal RNA profiles. So all these analyses may indicate that the nucleolus can act as a stress sensor during this desiccation loss. And another interesting finding is related to the accumulation of this molecule PAP, that is 3' phosphoadenosine 5-phosphate, which is highly accumulated during priming, only in prime seeds during the dryback. This molecule is often related to drought tolerance, and it may act in a retrograde. It's a member of a retrograde signaling pathway between nucleus and plastids. So if we are to schematize around the factors that induce loss of desiccation tolerance, we can see here that this can be conditioned by low levels of PAP, as well as high accumulation of reactive oxygen species, DNA damage, and ribosomal RNA instability. So building up on this efficiency of these seed quality hallmarks, we can now enrich these tools not only with genes but also with other more versatile, let's say, molecules like the ROS, PAP, and the 5.8S subunit of the ribosomal RNA. So another problem related to designing efficient priming protocols relate to the fact that these are not only seed and genotype specific, but also seed lot dependent. So the environmental condition during the formation of these genes also influence both the germination and the development of priming protocols. So within this in mind, our idea to use the gene expression profiles as indicators of seed quality and subsequently priming efficiency started quite a while back. In, for instance, 2010, we looked at the expression of OGG1 and the FPG genes involved in the removal of oxidized bases from the DNA. And what we saw is that during seed imbibition, this gene peaked in the moments when water uptake also peaked. While when we use polyethylene glycol to simulate a more controlled water uptake, we see that these genes are less expressed. So these results were corroborated this year by some of our colleagues from Poland that monitored the expression of these genes in barley, wheat, and rye, showing species-specific expression of these genes during imbibition, and that this also corresponded to water to the imbibition phases and the pace of water uptake. Then during in relation to priming, we evaluate this variation in a system looking at different varieties of different accession and different species of eggplants or solanum species. So here you can see comparatively how different hydropriming timing affect the efficiency of germination into different varieties. On one side, the cultivated accession of eggplant, and in the other side, wild type relative. So you can see in first case, the eggplant seeds take around 8 days to germinate, while after the application of hydropriming at different time points, we arrive to make them germinate even from the first day. So the 72 hours hydropriming pre-treatment was the most efficient. While in the case of this wild relative variety, we can see that treatments with 12 or 24 hours had a similar effect. So these results are part of a past project, a national project that aimed to characterize a vast germplasm collection that was present with our colleagues from the CREA Institute. So in this case, we evaluate all these germplasm collection, taking into consideration also the seed lots collected at different years. And based on the germination profiles, we have then grouped all these different accessions into early, normal, and late germination accessions. Then we went to see how the gene expression hallmarks are applying to this system, and we actually observe quite high variability and heterogeneity when comparing all these different accessions. But however, when we went to measure the levels of the reactive oxygen species, here we could easily distinguish between low quality and high quality seed lots, given that the low quality ones had higher accumulation of these molecules. So the last example that I want to present to you in this system is the latest study that we published this year together with our colleagues from the University of Milan that used two genotypes of maize, the low phytic acid mutant and its wild type counterpart. So even if the low phytic acid is a trait that is generally required because it enhances the mineral bioavailability during consumption, it has also some negative effects, especially on the germination and the viability of seeds during storage. So here we have applied hydropriming treatments following artificial aging, and we were able to show that the LPA mutant had improved germination up to 20%. Then, in this case, what we did is instead of looking at genes, we went back to look at the metabolites. And after mass spec analysis, we were able to show that the changes we observed were related both to the genotype as well as the priming treatments. I want to just catch your attention to some of these metabolites that are still polyamines, like the spermidine and the ornithine. You can see that they are present at a low level in the low phytic acid mutant, and their level highly increased after the hydropriming. So this proves us again that these type of molecules have a protective role, and they can be used as markers of seed quality even across different species. So to conclude, we saw how we went from basic models of the pre-germinative metabolism and the identification of key molecular players of hallmarks of seed germination and priming efficiency. Then we switched with the translational research. We switched to from model systems to crop and wild relatives, trying to use these hallmarks to evaluate the seed quality, as well as to propose tailored seed priming protocols. Nowadays, we are moving a bit towards a more, let's say, applied field, but this applied field that I'm speaking here, it's bit different than the industrial and commercial application that you are going to see with the next presentation. But anyhow, we are trying to apply this knowledge to other crops, like climate-ready crops, taking into consideration also sustainability and circular economy aspects. So within this case, we are having several projects, novel projects that were funded by the European Union or the local initiatives, starting from the Benefit Med project, coordinated by Alma Ballestrati, where we are developing tailored priming protocols for orphan legumes, and testing their efficiency not only in the lab or greenhouse, but also under open field conditions. While the Implantomics project led by the University of Sfax in Tunisia is proposing development of new facilities and training of personnel to look into different plant omics. And here we are targeting cereals, and our work in this relates to targeted priming treatments and the application of metabolomics. While at the national level, with the PNRR funds, we are applying plant-based biostimulants for the treatment of seeds, so applied during priming, and these biostimulants are actually obtained as plant extracts from agri-food waste. So to conclude, I'd like to thank all the members of our labs, starting from Alma and Andrea Pagano, and all our very highly motivated and dedicated PhD students, as well as our collaborators. For instance, Susanna Araujo was mainly involved in all the works on Medicago truncatula, our colleagues from University of Pavia, which are experts in biochemistry, chemistry or ultrastructural microscopy, our colleagues from Milan for the work on maize, and our colleagues from CREA for the work on eggplants. And obviously we have to thank all the financial support that we got from different local or European initiatives, as well as from some seed companies. And I'd like to thank you for your attention, and also take some time to thank all the international community for all the work carried out within this topic. Thank you.
Thank you very much, Anca. That was a very interesting presentation. We will have, after the next one, a few more questions, and we hope to have enough time to answer all the questions. Otherwise, they can come back to you directly as well. Just one question which came up was, do we have the presentations ready on somewhere to be looked at? Yes, that will be on the ISTA website, and the webinar will be also shown, recorded on the ISTA YouTube channel in a few days' time. You may also subscribe to both the ISTA and ISSS newsletters, and I posted the ISTA link in the chat for you. Now we are going to invite another colleague of ours, Dr. Tomoko Sakata, who has been working at Incotech for about 23 years now, which is a specialized company for seed enhancement technology in R&D, mainly for priming, and currently she is responsible for R&D of Asia Pacific region as the respective manager of that region. She has obtained her PhD from Chiba University in Japan and has experience in seed business sector for more than 30 years in Japan and in the Asia Pacific region. She started her career at a vegetable seed company in Japan and expanded her scope into the field of crop areas, especially to seed coating and to disinfection technology development on paddy. She's currently a member of the APSA, that's the Asia Pacific Seed Association Seed Technology Committee, and the ISTA Advanced Technology Committee. Her presentation is about seed priming, practical commercial use, and its evaluation. And so As our president, Keshavulukunsot, just mentioned at the beginning, that is bringing the practical way to the science. Tomoko, the floor is yours. We all will enjoy your presentation, I'm sure. Thank you very much. You're still muted, Tomoko.
Thank you very much. My presentation is visible? Okay, good.
Yes.
Can you hear me? And my presentation is visible?
We can hear and see very well.
Okay, very well. Thank you very much. Then, ladies and gentlemen, good morning, good afternoon, and good evening. I'm Tomoko Sakata, R&D Manager, Infotech, Asia Pacific, and ISTA Advanced Technology Committee member, and a member of the Standing Committee for Seed Technology in ISTA. Thank you very much for joining this webinar, and thanks for giving me such a special opportunity to discuss such interesting topics. The title of my session is Seed Planning, a Practical Commercial Use and its Evaluation. First, I'd like to discuss the general matter of planning, and after that, I'd like to move to the topics what germination test method is preferable to understand the priming effect more correctly and widely. Let me start my presentation. Then, first, I'd like to start my session from the discussion regarding what is priming and its practical use. What is priming? As everyone knows, priming is a technology to get a faster and uniform germination. Photo and the graph are showing the typical germination speed and the uniformity improvement by priming. However, not only germination acceleration, priming can relieve the dormancy and improve the germination under suboptimal condition. This is one of the big benefits. Therefore, we can say that priming is kind of insurance to get stable germination even unexpected climate condition happens. There are various priming effects such as repairing membrane and DNA, vernalization, etc. However, today I'd like to focus on the effect of priming to improve the germination speed, uniformity, and germination at a suboptimal condition. Here, I'd like to explain the principle of seed priming shortly. Water uptake by seed is triphasic, as described in this graph. If you plant a seed, it takes number of hours, days, and weeks before the first seed germinates. This is phase 1 and 2. And after finishing phase 1 and 2, germination happens. Image of right side is explaining the basic principle of priming. Priming is the technology to control the seed metabolism process, not to occur the germination, by controlling the moisture content of the seed and environmental factors, and stop the seed metabolism process by drying back the seed. This is the typical explanation of a priming process. Germination preparation stage was already done by priming process. Therefore, a primed seed can germinate faster after absorbing the moisture. However, nowadays, we can say that the definition of the word of priming is used to express a wider technique which can improve the germination, not only metabolism control. As already explained, benefit of priming is to get faster and more uniform germination. It leads to increased usable transplant more. This is a big benefit in nursery industry. In addition, it has effect to enhance the germination at suboptimal conditions, such as high and low temperature, drought, salinity condition. These effects are beneficial. In that these are the major benefits of priming in seed industry. Can you see my screen? Hello?
Yes, we can see your screen.
Okay.
We can
Okay.
see. It's.
Thank you very much.
Now it's Earth's average temperature showing on your screen.
Okay. Yeah, it's correct. This is
Okay.
the graph which explains the average temperature per month since 1815 to today. Average temperature has been clearly increasing year by year. And the data of this year is showing a big deviation from the trend in past. To tackle with such unexpected climate deviation, priming is very useful technology as insurance to get stable germination. This is the example of crop seed are primed in each region in commercial level in practical agriculture nowadays. Requirement of priming treatment are different per region due to the difference of genetic background, cultivation way, environmental challenges, seed price, and market demand of final product uniformity. However, we can say that priming is mainly used on vegetable seed generally due to the high seed price, which can accept the cost of priming. However, the demand of priming has been expanding in bulky crop and agronomic crop as well due to the requirement to get a better germination in suboptimal conditions. Good examples are sugar beet in Europe and cumin seed priming in Asia, especially in India. I'd like to share some example of new trend in priming. First, crop point of view. Nowadays, the demand of priming treatment has been raised in through potato seed in Europe. In Asia, priming demand on Asia-specific crop such as gourd, cucumber beets, and okra are quite hot. And tropical crops such as papaya are also expected. And due to the trend of organic food, the requirement of organic priming method has been increasing, especially in Europe and the US mainly, and slowly expanding in Brazil and Japan as well. To understand the benefit of priming, its evaluation is very, very important. Priming seed has different character from original raw seed, such as faster germination, good uniformity, better germination at suboptimal conditions, such as low temperature, high temperature, right condition, etc. However, ISTA germination test method is designed to evaluate the germination capacity at the best condition of each seed. A germination seed, bigger, uniformity are not targeted. Germination at a suboptimal condition is not targeted. Due to such reason, actually, it is challenging to evaluate the priming efficacy properly by ISTA germination test method. Then we need to consider what germination tests are required to evaluate the primed seed properly, and if we don't know the proper evaluation way of priming seed, we cannot use the full benefit of priming. I think many participants who relate with ISTA are working in quality control area and having interest in germination test method more than the priming process itself. So from now on, I'd like to discuss regarding the germination evaluation of primed seed. To evaluate the primed seed germination correctly, test per species, variety, and even per seed lot depend on the purpose of priming. For creating a nice test to evaluate the priming efficacy, the factor which need to be considered would be environmental condition where each seed are sown, the frequency of germination count to know the germination speed improvement, and what abiotic stress happen where each seed are sown. For example, lettuce. Lettuce is a very famous crop for its difficult nature in germination, such as summer dormancy. It always depends on the dormancy level of the seed. However, germination decrease happens generally above around 20 centigrade. Next is photodormancy. Seed has difficulty to germinate at a dark condition, although pellet is popular in lettuce seed due to its small seed size and difficult shape to handle. It means that the seed has a chance to be packed in darkness by pelleting in practical condition. And another challenge is in lettuce seed is secondary dormancy. ISTA suggests to perform the germination test on lettuce seed at 20 centigrade and count at 4 and 7 days after sowing. However, in this case, it is not possible to evaluate the dormancy level of raw seed, and priming effect improve the dormancy, even though the issue needs to be solved by priming its germination at higher temperature condition. Therefore, a germination evaluation at a wider temperature range in both of raw seed and primed seed would be very helpful, such as 16 to 36 centigrade to centigrade interval, and frequent germination count is suggested because raw seed can germinate within 1 day at optimal germination conditions such as 20 centigrade. And top of paper and soil test would be helpful to check the deterioration level. In addition, periodical repetition test is suggested due to the short shelf life nature of lettuce seed. Some gradient table is the equipment which can check the germination at a wider temperature range at the same time. Lettuce priming needs to have the effect to break the thermodormancy as well as photodormancy. Therefore, it would be useful if germination test is done at a wider temperature range using such equipment under the dark condition. Please find the image of right side. The table is set to create temperature gradient from 16 to 36 centigrade into centigrade interval. If seed is sown in each temperature zone in linear from left to right side of this table, then it is possible to evaluate the germination at such wider temperature range at the same time. Additionally, this test is possible to evaluate the photodormancy level because this table is put in green light room to be eliminate the light effect. In case of the temperature zone around 20 centigrade, which is suggested by ISTA on lettuce seed, it is quite difficult to know the dormancy level of raw seed and the dormancy breaking effect of priming. However, if germination test is done in wider temperature range, especially high temperature area, the dormancy level of raw seed and the priming effect to break the thermodormancy are clearly visible. This is the germination comparison between raw seed and primed seed at 3 different temperatures. This is showing what the germination difference happen, especially high temperature condition. Meanwhile, no difference is found in comfortable temperature zone around 20 centigrade. Next topic is the storability and shelf life of primed seed. Primed seed storability is influenced by the original raw seed quality. Here, I'd like to discuss the risk to apply priming on aged seed and low vigor seed, and highlight the importance of periodical repetition test, especially in lettuce seed. In this graph, a blue line is high vigor seed, and the red line is low vigor seed, and the solid line is raw seed, and the dotted line is primed seed. This graph is showing that low vigor seed also may have chance to show the some nice priming effect just after treatment. However, germination decrease may occur quickly. Meanwhile, high vigor primed seed keep longer shelf life. The photo in right side are showing the healthy germination and the typical abnormal germination caused by deterioration in lettuce. Those are germinated. However, cotyledon and root development are not proper. So I'd like to share some tips to use priming safely and stably while getting maximum benefit. Low vigor seed may show positive result initially, but will deteriorate sooner. Therefore, priming is not a technology for all the seed lot, nor low quality and low vigor seed. If you want to use a priming technology with good efficacy, I strongly suggest you to apply it on fresh and good quality seed only. To get the priming effect with good shelf life, optimization of the priming recipe per seed lot is required, not per variety and crop. And periodical repetition test is important, especially short shelf life seed or sensitive seed, such as lettuce. Okay. Detecting the risk of germination decrease in advance is very important in seed industry. Therefore, a periodical germination test would be helpful to check the increase of deterioration signs such as necrosis, symptom on cotyledon, and abnormal root, not only germination decrease. Next is tomato. Tomato seed has a chance to be sown in nursery for seedling production for grafting purpose. Therefore, faster and uniform germination is expected mainly. And if it can germinate faster at lower temperature, it is beneficial. ISTA is suggesting 20-30 centigrade alternated start counting from 5 days after sowing. However, germination test at 15 centigrade with earlier count, such as 3 days after sowing, would be useful to understand the priming effect clearly, as shown in this graph. This is the photo which took at 6 days after sowing at 20 centigrade condition. Priming effect is clearly visible by the size of plant. However, both of raw seed and primed seed are already germinated a lot. In this case, it is difficult to express the difference between raw seed and primed seed by number of the germinated seed. It would be valuable to get the data which can show the germination speed improvement clearly by starting the count earlier. This is germination test result of some gradient test at 10 to 30 centigrade temperature range. The graph in left side is the 3 days after sowing count. This is showing the germination difference between raw seed and primed seed clearly in all temperature range. The graph in right side is 14 days after sowing count, which ISTA suggested as final count. As you can see, germination difference is not visible anymore above 20 centigrade condition. However, at lower temperature zone, we can see the difference still. This is a movie which compared the germination of raw seed and primed seed at top of paper and soil. The difference is very clear. This is the germination comparison of the interspecies hybrid type of rootstock tomato, which takes long time, such as 21 to 28 days to get full germination. The priming benefit is very clear by this video. This is the germination test at the nursery environment. Such test as real condition is also very helpful to understand the priming performance at practical use. Next is watermelon. Such tropical crop can germinate at higher temperature condition very fast. However, in lower temperature condition, germination gets slower and in some cases germination decrease happened. If priming can work to improve the germination at lower temperature condition, it is very beneficial in seed industry. ISTA is suggesting 20 to 30 centigrade alternate temperature condition, starting the count from 5 days. However, lower temperature such as 15 to 25 alternate temperature setting with earlier count would be valuable to evaluate the priming effect for practical use. This graph is explaining the value to test the primed watermelon seed at lower temperature to understand its benefit clearer. Next example is bitter gourd. This is the same story with watermelon. However, this is more remarkable example. Germination at lower temperature is challenging due to the tropical crop, although it has a chance to be sown in winter season as well in Asia. So, germination tests at lower temperature, such as 15 to 25 centigrade alternate temperature setting, would be valuable to evaluate the priming effect, whether it meets to the seed industry requirement. Here, I'd like to share the example of germination test comparison in bitter gourd. Germination test at 20-30 alternate temperature condition, primed seed reached to the maximum germination around day 10, and it is 3 days earlier than raw seed. You may think that 3 days germination speed improvement is not so valuable. However, in 15-25 centigrade alternate temperature condition, raw seed start germination around 20 days after sowing. It is 10 days slower than a higher temperature condition. In this condition, germination acceleration effect by priming is more than 7 days. We can say that this testing method is realistic if farmer is expecting the germination improvement at lower temperature condition in winter season, such as India. Next example is okra. Okra is challenging in germination due to hard seed coat. In addition, germination at lower temperature condition is difficult, although it has chance to be sown in winter season in Asia. Therefore, germination test at lower temperature, such as 15 centigrade, would be variable, and area count is variable to know the germination uniformity. At 25 centigrade condition, both of raw seed and primed seed germinate very quickly, and it looks that priming is not so variable. However, at 20 centigrade condition, raw seed germinate get lower, and at 15 centigrade, it becomes more significant, even though primed seed is still showing higher germination. This is one of the remarkable examples which explain the necessity of alternative germination test to evaluate the real benefit of priming. Last example is papaya. Papaya seed need long time for germination. It is around 21 to 28 days. Therefore, germination improvement with good uniformity is required in seed industry and nursery business. If germination count is done earlier, a priming benefit would be clearer. ISTA suggests to do the count at 12 days and 28 days after sowing. However, to know the germination uniformity, the frequent germination count between 12 to 20 days after sowing would be interesting to check. Then I'd like to move to the summary of this session. Priming with the technology to make germination faster, improve the germination at suboptimal condition. Therefore, to know the germination speed improvement by priming clearly, it is suggested to start germination count earlier, more frequent germination count, good germination rate and uniformity test, 2 count per day to get exact description of germination speed and uniformity. And the time-lapse video is also helpful to know the difference visually. And to evaluate the germination improvement by priming at a suboptimal condition, some gradient test, moisture stress test, different light condition, temperature stress testing at low and high temperature would be variable. To know the impact of priming on seedling quality, plant type test to check the cotyledon condition, usable plant transplant test, test based on the plant laser evaluation to determine the percentage of seedling that can be transplanted. Such tests are variable. And to know the impact of priming on shelf life, storage test, checking the germination periodically, is very important in seed industry. Most important things for designing the optimal germination test method to evaluate the primed seed performance and its value are to understand germination character of each seed and to understand what priming effect you are expecting. Special thanks to the R&D team in Incotec Asia Pacific and Dr. Henry Wilkin in Incotec Europe, who gave me the nice advice to make this presentation, and all of my fantastic colleagues in Incotec. Thank you very much. And all data, photo, information in this presentation is from Incotec. Thank you very much. Thank you very much for your attention, and any questions are welcomed.
Yes, thank you very much, Tomoko. We also ask Anca now again in the in video here. Very, very good presentations. I think we have time for at least 1 or 2 questions to all of you, which are came in from the audience. All the others will be forwarded to you, and you can answer them. There was one, maybe Tomoko, we start with you. We have difficulty in getting priming right with soybean seed. Even in precondition, we still see a lot of damage due to inhibition. Is there any protocol that we can apply? And you're smiling, so you will be telling us you don't have an answer.
Yeah, actually, the soybean is very challenging. If it absorbs the moisture, usually get damage, and the germination decrease also happen. So unfortunately, I don't have nice priming protocol for soybean at this moment. However, I know such a requirement exists in this industry. So in the future, I want to consider to develop somehow. But at this moment, sorry, I don't have nice idea for that.
Okay, there was another one coming for soybean as well, but I think we answered that one. Maybe another quick one to you, Tomoko. How are seed-associated pests managed during priming?
Pest manage.
Yes.
Pest, not a disease, pest.
Maybe some fungi on the seed. Do they have any influence?
Yes. If seed moisture is not controlled properly, maybe such fungus disease or bacterial disease increase happen. So priming process need to be managed very clean, and after that, after drying, seed moisture need to be returned to the nice condition. If seed moisture is higher, of course, seed have risk to get such infestation and increase during the storage. So moisture control management during the process. Process need to be clean, and after priming, seed need to be dry back to the optimal moisture level. That is the key to control the disease and the fungus.
Thank you very much. Anca, I've got 1 or 2 questions also for you. One is coming in. When you talk about over-priming, what does that exactly mean? Does it mean that seeds during priming start to germinate?
Sometimes, yes, sometimes radical protrusion come out, but yes, the idea is to go over the indicated time. Like if you think that the inhibition curve, let's say in the case of Medicago truncatula, already at between 8 and 12 hours, you are already in the plateau, and seeds start to germinate after 18 hours already, having the little radical come out. So if you dry them back, after putting them to germinate again, the seedlings are grow a bit of aerial part, but no roots. So yes, over priming means that you may arrive to the radical protrusion, and the treatment is too long. It's too long.
Mm-hmm. Okay, maybe here's another one. Between the environmental and genetic factors, which affect more seed quality and involved in seed dormancy?
Seed dormancy, we are not looking much into it at the moment, but for sure there is both the genetic and the environment, and as well the interaction between them that are affecting both dormancy, germination, and everything. The tools that we applied so far, we did not yet look into the dormancy part. They are all non-dormant.
Yeah, thank you very much for your
later.
Yeah, thank you very much. All the others, other questions will be answered later. I have a thank you here also from the president of ISSS, Julia. And as she's mentioning, it was a very nice and excellent presentation, which I can confirm because I got other requests back from the audience. Very interesting. I think, Keshavulu, you may want to thank as well. You can join us on the screen if you like. You're still muted, Keshavulu.
Thanks, Andreas. So fantastic presentations from both the speakers. So one could cover the molecular aspects of seed priming, and the next pre-generative aspects of pre-germinative methods and then aspects. So that is the most important part for the fundamental is knowledge to the those who are practicing and those who are in the research areas. And then coming to another important topic which we could hear from again Dr. Tomoko, and then it's a practical usage and how these seed priming techniques are useful, and then what are these effects, and then what are the differences between the primed and unprimed seeds. And then she narrated and she showed with the figures, and really it's a great presentation from both the speakers. Then coming to I think so I have there are the seed priming is of course we should always consider this is one of the promising tool for the sustainable agriculture, and also and the climatic, and then climate again the important, and then in the climatic climate, to address the climate challenge also this technique is the important one. But however, we need to integrate this technique with the agriculture practice. So then only then, so farmers or seed industry can able to start, and then so they can able to practice this technique. And then so, one or two the important aspects given by the Dr. Tomoko. She also gave these primed techniques can reduce the germination duration, and then it gives a faster growth and faster field emergence, and also it gives a good field performance or seed performance under suboptimal conditions. So that is important, especially in the regions of the tropical and subtropical situations. So therefore, she gave us the crop-wise, the vegetables mostly, and also the papaya. And then so one of the attendees, they have asked, he asked about the soybean. I think so. It's the soybean, and again the old seeded crop, and then it is impossible to again imagine the seed priming in soybean, especially. So as per my knowledge, it is again. So therefore, I think so. There are certain future studies are required for the field crops as compared to the vegetable crop. The seed priming research or seed priming practices are less in the field crops or gold seeded crop species. So therefore, it's a gray, and then so the challenges are ahead, especially in the gold seeded crops. Despite these challenges, so this technique is a very valuable for improving the seed germination, field emergence, and then the crop performance as a result. Yeah, for the farmer, especially he improves the productivity and then production. So thank you very much for the both speakers. I would also thank Andreas for the moderating this wonderful webinar. Definitely, so it is useful to the seed testing laboratories and then more of the seed industry. So those who are thinking in the areas of the seed priming. So thanks once again, and thanks to all for all the attendees for this webinar. Thank you very much.
Thank you, Keshavulu, and thank you again to the presenters. And just a reminder for all of you that the data will be available on the ISTA website. You have the link in the chat box, and also on the ISTA YouTube channel, you will find this webinar in a few days' time when it is downloaded and uploaded there. There will be future topics for this ISTA-ISSS webinars, and we will continue to do them in the next year. We plan to, like the past years, and as Keshavulu, you mentioned, it was the number 6 now, so we are in the end of the 3rd year of this session, and the audience tells us that it is well received and that it is it's very good. You will find all the news on the newsletters of both organizations, and if you go to the website either of ISSS or ISTA, you can register to the newsletter and get the information when the next webinars are coming up. I do thank you very much, and we wish you all the best for the rest of the year, and for those who we don't see, it's very early, but have a good holiday time. Thank you very much. Goodbye. Bye-bye.
Thank you, Ananda. Thanks. Bye-bye.
Thank you very much.